Hello students, in this video we are going to discuss the topic bacteria of botany. Okay, so let's discuss from the very basic of the bacteria chapter. First of all, we all know that there are five kingdom classification in a biology. Okay, so five kingdoms are Monera, Protista, Fungi, Plantae and Animalia. So out of these five kingdoms, the Monera is included in the prokaryotes and all the other kingdoms are included in the eukaryotes. Okay, so now what's the difference between prokaryotes and eukaryotes? Let's discuss. So prokaryotic cells mainly doesn't contain any membrane bound organelle. Okay, so there will be no membrane bound organelles like endoplasmic reticulum, Golgi body, or mitochondria, etc. etc. And especially there will be no membrane around the nucleus. Okay, so there will be no nuclear membrane, so there will be just chromatin. Okay, and whereas in the eukaryotic cells all the structures are present. You can see here there is nucleus, there is Golgi apparatus, there is endoplasmic reticulum, there is vesicles, etc. etc. Okay, so this is the main difference. So main difference is prokaryotic cell doesn't contain membrane bound organelles while eukaryotic cells contain all of that. Next is prokaryote. We have discussed that Monera is included in the prokaryotes, and Monera, the kingdom Monera is basically consists of the bacterias. Okay, so bacteria, we can say that bacteria may be of two types, eubacteria and archaebacteria. Eubacteria means true bacteria and archaebacteria means those bacteria which are found in extreme conditions like very hot temperature or very cold temperature. So these are called archaebacteria. So bacteria are discovered by obviously Anton von Leeuwenhoek. So if we talk about the main characteristics of the bacteria, so obviously it's the prokaryotes, so there will be no membrane bound organelle. Next main thing is the cell wall of the bacteria consists of the NAG and NAN, N-acetyl glucosamine and N-acetyl muramic acid. We can say it peptidoglycan or murine. Okay, so this is the point you have to remember always. And as the bacteria is prokaryote, so ribosomes present in the bacteria will be of 70S type. Okay, you should know that ribosomes, there are two types of ribosomes. One is 70S and one is 80S. So in prokaryotes, ribosomes are 70S type, and in eukaryotes there will be 80S type of ribosomes. Okay, so next thing is let's discuss about a staining method that is called Gram staining method, developed by the Hans Christian Gram. So what he did basically was he put some bacteria on the plate and then he applied a basic dye named the crystal violet, and this dye basically gives the purple color to all the bacterial cells. After applying this dye, he applied iodine. Iodine is used as a mordant to increase the affinity of the stain. Okay, after using iodine, he decolorized the cells with the alcohol. After decolorizing with alcohol, he applied the safranin dye, that is another dye which gives orange color, or you can see here that pink color. Okay, so after completing this process, what he observed in the end was there were two types of bacteria. One type was of purple color, okay, and another one was of pink color. Yani some bacteria retained the purple color which he applied at the first, but some bacteria decolorized after alcohol wash and gained the another dye that is called safranin. So he concluded that there are two types of bacteria, and the bacteria which retained the purple color, he named it as a gram-positive bacteria, and the bacteria which became decolorized after alcohol wash, he named it as gram-negative bacteria. Okay, so there are two types of bacteria, gram-positive, gram-negative, according to this staining method. Okay, but what was the basic difference between the gram-positive and gram-negative bacteria? So the basic difference between these two bacteria is the difference between the cell wall. What was in the gram-positive bacteria? There was obviously a plasma membrane, and then there was the thick cell wall over that plasma membrane. Okay, same, but there was a little bit difference in the cell wall of the gram-negative bacteria. There was plasma membrane. After that, there was cell wall, but it was thin, not thick as much as of gram-positive bacteria. So it was thin cell wall, and after the cell wall, there was extra layer that was contained of the lipopolysaccharide. So lipopolysaccharide outer layer called as outer membrane. So outer membrane that was more permeable than the cell wall. So this was the difference between the cell wall of both bacteria. And the outer membrane of the gram-negative bacteria contains various proteins named porins. The function of these porins were to move the substance in and out across the membrane. Okay, so it was basically more permeable, and that's why we saw that after decolorization with alcohol, gram-negative bacteria easily removed the purple color and easily gained the safranin dye. That's because the outer membrane of these bacteria were more permeable. Okay, so that was the difference. So main difference, if we talk about the main difference between gram-positive bacteria and gram-negative bacteria, then it will be the difference in the physical properties of their cell wall. Okay, so move to the next. If we talk about the some special structures of the bacterial cells, you can see here there are many small hair-like structures all over the bacterial cell that is called fimbriae. Okay, so these fimbriae are used for the movement of the bacterial cells. Understood? For the movement of bacterial cell itself, and in the attachment of it to the surface. Next special structure is pili. Pili are the same hair-like structures like the fimbriae, but they are not as many as fimbriae, but and their function is to help in the sexual reproduction of the bacterial cell. Okay, so pili will help the bacterial cell in conjugation process. So they are basically called as the sex pili. Next special structure in the bacterial cell is nucleoid. As we have discussed earlier that bacterial cell doesn't contain any nuclear membrane. So if there will be no nuclear membrane, then there will be obviously just a thread-like structure called chromatin. Okay, without any envelope or membrane. So that thread-like structure, which is placed in a separate place, that is called nucleoid. Okay, so in the bacterial cell there is not nucleus, but there is nucleoid. So it is the main DNA of the bacterial cell called nucleoid. Next special structure in bacterial cells are called as plasmids. Now plasmids are the additional circular DNA. Okay, so these are the different from the main DNA. So these are additional circular DNA present in the bacterial cells. Now what's their function? Their functions depends on the genes which they carry. Okay, sometimes they help in reproduction, sometimes they provide resistance to the bacterial cells, And on the basis of these functions, plasmids are of three types. We can see here F plasmid, that is responsible for the transfer of genetic material, R plasmid, that carry genes for the resistance to antibiotics, and O plasmid, that carry genes for the production of a protein named colicin, and which kills the sensitive E. coli cells. Okay, so these are the plasmid. In the last, I'm gonna share you some very important thing, that is, we have discussed about the gram positive and gram negative bacteria, right? So you can learn two examples of the gram positive and gram negative bacteria. So the two gram positive bacteria which you can learn and which are very simple to learn are, first is Diplococcus pneumoniae, okay, and next is Salmonella typhi. Diplococcus pneumoniae is a bacteria which cause the disease called pneumonia. Okay, so from pneumonia you can learn Diplococcus pneumoniae. And Salmonella typhi is a bacteria which cause typhoid fever. Okay, so from typhoid you can learn Salmonella typhi. So these are two gram positive bacteria. And what about gram negative bacteria? First is E. coli. E. coli is a bacteria, and next is Vibrio cholera. Both these bacteria are related to our intestine. E. coli is a bacteria which is found in intestine of human, okay? And next is the Vibrio cholera. It's a bacteria which cause the disease cholera, which is also related to intestine. So these are two examples of gram negative bacteria. Thank you.